The goal of this procedure is to induce and observe autophagy in a filamentous fungus. This is accomplished by first inoculating small volumes of nutrient-containing medium with spores or small mycelium pieces. In the second step, starvation pads are prepared in the cavities of glass microscope slides, and then the inoculate is transferred onto the pads. In the final step, the starvation pads are incubated in a wet chamber to induce autophagy. Ultimately, the autophagy can be tracked by monitoring the translocation of fluorescent marker proteins of interest to the vacuoles using fluorescence microscopy.
This method can help to study key issues in the cell biology field, such as identification of new compounds that modulate the autophagy process.
To prepare for a starvation experiment, if the P. chrysogenum strain of interest is kept on green rice, first place 2 to 3 rice grains covered with sporulating mycelia into a 1.5 microcentrifuge tube, and fill the tube with 500 microliters of yeast extract glucose growth or YGG medium. Then vortex the tube for 30 seconds so that spores detach from the rice, and incubate the tube for 1 day at room temperature. The next day, dilute the spore suspension to a 1 to 50 concentration in sterile tap water and mix well. If the P. chrysogenum is kept on agar, use a pipette tip to transfer small pieces of the mycelium into a 1.5 milliliter microcentrifuge tube containing